Hello everyone, so I'm making a video on microbiology second year record. Let's start. Firstly, we will go to the contents. These are the contents. So the first one is microscope. So to study various parts and operation of microscope, the principle and types of microscope. So whatever I have included here is being taught, and yeah, so this is the picture I've given and the parts. So here is the components of a microscope, of simple microscope. And this is the care of microscope. So the second one is hanging drop method. So why do we do hanging drop method? To examine motility and identification of unstained organisms in a given specimen or culture. So to examine motility mainly, so identification is done for unstained organisms. So here is the procedure I've drawn, and these are the requirements and the principle, and this is the procedure. So coming to the simple staining, to perform simple staining procedure, yeah, why do we do? To compare morphological shapes and arrangement of bacteria. So here is the specimen, and this when we inoculate on culture media. So when we do simple staining, we can see the morphology and the arrangement of bacteria. So these are the requirements, principle and procedure. So coming to the gram staining, so it is used to study morphological appearance of bacteria and the differentiate between gram positive and gram negative bacteria. So what's the principle? Here we are using 4 types of stains. So primary staining, modern alcohol or acetone treatment, and counter stain. So this is the procedure of gram staining. When we take a specimen and inoculate on a culture media after incubation, so when we do a gram staining, we can see the gram positive cocaine chains and gram positive cocaine groups. So rods or bacilli, gram negative, and gram negative cocaine pairs, diplococaine. So these are the requirements, reagents to be used, and this is the procedure. Yeah. So in your practical examination, so be prepare especially gram staining, the principle. So coming to the Ziehl-Neelsen staining, yeah, why do we do Ziehl-Neelsen staining? So here is the method of Ziehl-Neelsen staining. A specimen when we inoculate, take on a slide and do Ziehl-Neelsen staining, we can see the acid fast bacillus, pink colored, bluish background. So be prepared for these slides when you are given for the practicals. So you will be given all these slides and you will be gone through for that. So yeah, these are all the viva questions. Be prepared for that. And why are we using 20% H2SO4? For which bacteria we are using 5% H2SO4? So this is the procedure. And yeah, here 5% H2SO4 is used for Mycobacterium leprae. And yeah, be prepared for that. So coming to the capsule staining, to demonstrate capsule of bacteria using capsular stain. So this is the principle and these are the requirements. Yeah, these are the reagents to be used. So this is a picture I've given of capsule here. So pneumococci. Here is the procedure. So coming to the spore staining, to determine the given spores of bacteria by using malachite green. So here is the procedure. The specimen, when we are inoculating on a culture media and incubating, then when we do spore staining, so we can see the spores. So coming to the flagellar staining, so flagella of motile bacteria. So principle, procedure, and yeah. So I've given few pictures of flagellas, monotrichous at one end of bacteria, and both the ends amphitrichous, one end many flagellas, lophotrichous, all the end, all over the bacteria, peritrichous. So coming to the culture media, I have given here the composition and uses of solid media, and yeah, peptone water, glucose broth, and nutrient agar plate. So coming to the culture media's preparation of media. So here is the preparation of media. How do we prepare? So solid media, liquid media, and semi-solid media. So composition and uses of liquid media. So blood agar and chocolate agar plates, these are. So yeah. Here is I have described about differential media. Example, McConkey agar medium. So yeah, there is a composition of this media. So next is selective media, transport media, anaerobic media, then enrichment media, sugar media. So his serum sugars for pneumococcus. So be thoroughly prepared for the composition and uses of medias. So yeah, you'll be placed plates and broths, and you'll be asked for identification in your examination. And yeah, whatever is being taught in your practicals, you just note everything, and you can so, and whenever you are provided with the plates, so you make a note of that. So you'll be getting confused between McConkey agar and the plates which have been placed to you. So yeah, be prepared for that, and practice thoroughly. So when coming to the stains, you will be placed slide, and you'll be asked for identification of whether gram positive or gram negative bacteria, or yeah, you'll be asked for that. So yeah, so coming to the inoculation methods. So to inoculate specimen or a bacterial suspension in media. So yeah, this is the principle, and I have given a picture of specimen. This is the specimen and this is the media. Yeah, sterile inoculated loop should be used. So specimen is inoculated into the media. So this is the primary inoculation. Here is the, yeah, how do we streak on solid medium. So methods include broth culture, streak plate, spread plate. So this is the spread plate. Pour plate, stroke culture, lawn carpet culture, subculture. So yeah, so this lawn carpet culture is useful for bacteriophage typing and antibiotic sensitivity. So yeah, be thorough with all the methods. I have given a picture here. This is the swab plate. So coming to the anaerobic culture methods, here I have described about to, to know and use of anaerobic culture methods. This is the principle and requirements, and then methods for cultivation and medias to be used for this method is described here. This is the gas pack system picture I have given here. So coming to the antibiotic sensitivity test, why do we do? To detect resistance and susceptibility of bacterial culture bacteria towards various drugs. So this is the principle. This is a picture of Stokes disk diffusion method and modified Stokes disk diffusion method. Principle, requirements, reagents to be used, and the procedure for Stokes disk diffusion method, and the Kirby-Bever disk diffusion method. So this is the picture for Kirby-Bever disk diffusion method. So I am going to make a video for rest well contents as a part 2. Otherwise the video would be very vast. And yeah, I'm making these videos for BSc MLT students. So make clear notes whenever you have practical microbiology practicals, and yeah, be you would be asked viva questions from, yeah, mainly stains, gram stain. You prepare thoroughly for that, and types of microscopes and the uses, all the medias, solid and liquid and semi-solid, and the uses, Ziehl-Neelsen staining and inoculation methods. I hope this video would be helpful for you to write your microbiology record. All the best for your examination. Thank you.